I'm going to be talking about excessive microglial phagocytosis contributing to neurodegeneration. And the main people who contributed to this were Stefan Milde, Amaj Puig-Delivol, Anna Villalta, David Allendorf, Claire Butler, and Alma Popescu. So you'll know that microglia are resident brain macrophages that specialize in phagocytosing things, extracellular things. So microglia can eat excess synapses and neurons during development to shape neuronal networks. So that includes eating live synapses and live neurons. And in the mature brain, microglia can protect the brain by eating pathogens such as bacteria and eating amyloids such as beta amyloid plaques, dead cells, and debris in order to protect the brain. However, excessive microglial phagocytosis of synapses and neurons may contribute to neurodegeneration, and that's what I'm going to be talking about today. So I'll just change the pointer here to a laser pointer. So it's known that microglia can eat synapses, that's both pre and postsynaptic components of synapses, and also axons, and also the kind of wrapping of axon, that's eating the myelin that wraps the axons. And microglia can also eat dendrites, but they can also eat the neuronal soma, which results obviously in neuronal death and loss. So we don't actually know a great deal about the differences between regulation of synaptic loss or neuronal loss or dendritic loss. But we do know a variety of factors that regulate microglial phagocytosis generally, and that includes fine signals. For example, ADP can be released from stressed or dying neurons and recruit microglia through the P2Y12 receptor. And similarly, fractalkine released from synapses and neurons can recruit through the fractalkine receptor on microglia. And there are so-called eat-me signals, such as the exposure of phosphatidylserine on the surface of synapses or neurons, which can result in microglial phagocytosis of them, either directly through phagocytic receptors on microglia that bind directly to phosphatidylserine, such as TREM2, or via binding of opsonins to the phosphatidylserine. For example, Gas6 can bind to expose phosphatidylserine and then bridge to the tyrosine kinase receptor, which is a phagocytic receptor. But there are other opsonins such as ApoE or MFG8 or C1q that can bind to phosphatidylserine and recruit other phagocytic receptors. There's also a soluble eat-me signal, UDP, which I'm going to be talking more about in this talk, which can be released from stressed neurons and induce phagocytosis through the P2Y6 receptor on microglia. Although this is more a kind of on a late phase of phagocytosis, the actual engulfment step. And there's also decylated cell surface, which is a major eat-me signal on many different cells. So most cells are highly sialated, so they have glycoproteins and glycolipids, which contain short sugar chains, and the terminal sugar of those short sugar chains is normally the sugar sialic acid. But that can be removed, for example, by sialidases, revealing sugars below that. So the first one is normally galactose residues, which is then bound by opsonins such as galectin-3, chiroticinin, or C1q, which then can recruit phagocytosis through various phagocytic receptors. But sialic acid is also a don't eat me signal, blocking phagocytosis of synapses or neurons by engaging C-clear receptors, which are basically receptors on phagocytes such as microglia, which inhibit, most of which inhibit phagocytosis. So sialic acid can act as a don't eat me signal. Also, CD47 is a protein which acts as a don't eat me signal by engaging SERP alpha on microglia, which inhibits phagocytosis. There are a variety of other factors, and it's probably a lot more complicated than that, but these are some of the key molecules regulating phagocytosis of synapses and neurons. So we're interested in this because of the possibility that neurodegeneration and brain atrophy may be mediated by microglial phagocytosis of brain tissue and neurons. And many neurodegenerative diseases involve brain atrophy, and brain atrophy occurs without significant neuronal death. That is, dead neurons there's not an increase in the number of dead neurons seen in neurodegeneration. There's just a neuronal loss. And there's little evidence that preventing neuronal death, for example, by blocking apoptosis, prevents neurodegeneration. And therefore, there's a possibility that neurodegeneration may be due to microglial phagocytosis of stressed but viable tissue. That is, neurons are lost prior to actual death. And that's of interest because this neuronal loss is at a relatively late stage in neurodegeneration. So this is just a time course of various things happening in Alzheimer's. And we know that amyloid plaques form 10 to 20 years before diagnosis of Alzheimer's. This would be, this line's indicating diagnosis of Alzheimer's, and this line's indicating diagnosis of mild cognitive impairment. And tau tangles occurs later, but again is a few years before dementia symptoms. But neuronal loss is a relatively late characteristic of neurodegeneration, and it's known that neuronal loss correlates with dementia better than plaques and tangles. And that's interesting because neuronal loss is not complete by the time of diagnosis of Alzheimer's. And therefore, the possibility is that post-diagnosis, one may be able to, if one could block the neuronal loss, one may be able to slow progression of dementia post-diagnosis. And so these neurons are being lost decades after they've been, sitting embedded in an amyloid saturated brain, and probably a few years after they've acquired tau tangles. So if you could delay the loss of those neurons by a couple of years, that would have a large effect on dementia prevalence, or at least that's the idea. Now, we know that microglial phagocytosis is associated with Alzheimer's disease from GWAS studies, and this is just a number of genes that are connected to microglial phagocytosis that are associated with Alzheimer's risk by GWAS studies. And this is just a review from Alison Goethe and colleagues identifying a variety of those genes and showing that they are preferentially expressed in microglia. So this includes things like clusterin, which is an opsinin. ApoE is also an opsin. That is something that extracellular, which binds to cells and encourages phagocytes to eat them. But we also have in here TREM2, which is a phagocytic receptor, a variety of receptors which inhibit phagocytosis, such as CR1, CD33, PILAR alpha, some genes such as phospholipase G gamma 2, which couple the phagocytic receptors to phagocytosis. And then there's genes such as ABI3, which regulate the cytoskeletal changes that occur in phagocytosis. And then there's a whole variety of genes such as BIN1 or PCALM, which are known to affect endolysosomal trafficking in one way or another. So there's a whole variety of genes associated with microglial phagocytosis affect Alzheimer's risk. And we also know that a variety of phagocytic genes are upregulated in the microglia of Alzheimer's patients, including TREM2 and its adapter protein DARP12, some of the opsonins such as ApoE and C1q, and a variety of phagocytic receptors are upregulated on microglia in Alzheimer's brains. However, this type of evidence doesn't clarify whether microglial phagocytosis is good or bad, or whether, for example, it's good in the early phases of Alzheimer's by clearing debris and amyloid, but bad later on by encouraging phagocytosis of synapses and neurons. So we need more evidence as to whether microglial phagocytosis is good or bad in Alzheimer's and other neurodegenerative diseases such as Parkinson's. So I'm going to start by talking about one of these genes, TREM2. So one copy of R47H TREM2 increases AD risk about fourfold. And normally that, because it's just one copy, that would suggest that it's a gain of function. However, there have been many different reports suggesting that R47H TREM2 is a loss of function due to reduced ligand binding. However, some of these reports are compromised by expression in mice, where it's known that R47H is misspliced, whereas it's not misspliced in humans, or the use of unphysiological ligands. So we decided to reinvestigate this as part of the PHAGO consortium. So the PHAGO consortium was a European Union IMI2 consortium of academic and commercial pharma labs across Europe. And in this case, we're looking at whether R47H TREM2 is gain or loss of function in relation to activation by phosphatidylserine. And this really was an international collaboration, in that the liposomes that were used here were developed by Sanofi in France as optimum ligands for TREM2. So that's a physiological mixture of lipids from the plasma membrane at reasonably physiological ratios of phosphatidylserine to phosphatidylcholine and cholesterol. And they developed these liposomes to be optimal ligands for TREM2. And then, so this was using human iPS-derived microglia, which were derived by AbbVie in Germany, and then were tested by Axam in Italy for activation of TREM2 by alpha ELISA, measuring phospho-Syk activation by these liposomes. And it was found that the R47H iPS-derived microglia then had a threefold increased activation of phospho-Syk by these liposomes compared to the wild-type or common variant of TREM2. So this is an opposite to the idea that R47H TREM2 is a loss of function. There was greater activation by phosphatidylserine liposomes in the R47H compared to the wild-type. The phosphatidylcholine cholesterol liposomes didn't activate without phosphatidylserine, didn't activate the TREM2 at all, and there was roughly equal activation by anti-TREM2 antibodies of R47H and the wild type. So this led us to then also look at phagocytosis of phosphatidylserine expressing targets to compare the phagocytosis in wild type and R47H. And this panel on the left here is using human iPS-derived microglia derived by Jennifer Pocock's lab in London, using for cells expressing the common variant or heterozygous for R47H. And we found an increased phagocytosis of synaptosomes. So that is isolated synapses, which did have phosphatidylserine on the surface. So again, this is an increase in phagocytosis in the R47H expressing cells. But these human iPS cells may differ in other ways. So we wanted to just isolate the effect of TREM2, and therefore we expressed the common variant, or R47H TREM2, in a human microglial cell line. That's Kymie3 cells, which naturally express a low level of TREM2, and we either express the wild type TREM2 or R47H. And again, the R47H expressing microglia had increased phagocytosis of isolated synaptosomes. They also had increased phagocytosis of phosphatidylserine coated beads. We then co-cultured these TREM2 expressing Kymie3 microglia with Lumis. Lumis is a human derived neuronal cell line. So these were co-cultured with the microglia, and we looked at phagocytosis and neuronal loss. And this is just quantifying neuronal loss when the Lumis cells are co-cultured with Kymie3 either expressing wild type or R47H TREM2, and there was increased loss when co-cultured with R47H TREM2. And overall, this suggests that R47H TREM2 may increase Alzheimer's risk by increasing microglial phagocytosis of phosphatidylserine exposed synapses or neurons. I should say that this story is complicated by the fact that TREM2 also inhibits background phagocytosis via TREM2 inducing cystatin F within the microglia. But if you want to know that story, just look at this recently published paper. So we showed a number of years ago, we published a series of papers about 10 years ago showing that if you inflammatory activate microglia with, for example, LPS or A beta or TNF alpha, this results in a slow progressive loss of neurons due to microglial phagocytosis of live neurons. And we investigated a variety of mechanisms involved, and that included the production of reactive oxygen species, which stress the neurons, causing a reversible exposure of phosphatidylserine on the neurons. So these neurons can reversibly expose phosphatidylserine as a result of calcium elevation or reactive oxygen species. And that encourages microglia to phagocytose those phosphatidylserine exposed neurons. And in part, that's mediated by binding of the opsin in MFG8, which then bridges between exposed phosphatidylserine and the vitronectin receptor, which is a phagocytic receptor that can induce microglial phagocytosis of neurons. But more recently, we've shown that that changes to microglia are also important in this microglial phagocytosis of neurons. In particular, inflammatory activated microglia become desialated, and that increases microglial phagocytosis. This is work largely done by David Allendorf in the lab, but he's shown that inflammatory activating microglia with, for example, LPS or tau or A beta or PMA and a variety of other things causes desialation of the surface of the microglia. So that involves removing the terminal sialic acid residues, and that's due to the translocation of a sialidase known as neuramidase 1 from the microglial lysosome to the cell's surface, and indeed it's released into the medium. And that results in desialation of the cell surface. That's a removal of these sialic acid residues. And that also occurs to a variety of receptors such as TREM2 and MERTK and TLR4. And David's shown that that can activate some of these receptors such as TLR4. I should explain that it's been shown previously that a whole variety of different receptors are inhibited by sialation, and if you remove those sialic acid residues, you can activate a variety of different receptors. But David showed that this was true for, and others have shown that's true for MERTK and TLR4 and for complement receptor 3. So I'm just going to show you a small amount of evidence from that. So what we're looking at here is LPS-induced neuronal loss. So this is using primary cultures of rats or mouse neurons and glial cells, including microglia. And we're quantifying the neuronal densities 3 days after adding LPS or not adding LPS. And you can see that 3 days later, LPS induces a loss of about 20% of the neurons. But if you inhibit extracellular sialidases with DANA, you can prevent that neuronal loss. So DANA is a sialic acid derivative, which can inhibit extracellular sialidases, such as neuramidase 1 when it's in the outside of the cells. And that prevents the inflammatory neuronal loss, and also prevents neuronal loss induced by A beta. And he also showed that blocking that desylation activates CR3 in terms of binding to ligands. And if you block the complement receptor 3 with a function blocking antibody, then you can prevent neuronal loss induced by LPS. But if you just desylate the surface of the synapse of the microglia by, for example, adding extracellular sialidase to remove these terminal sialic acid residues, you also get a neuronal loss, and that can also be prevented by blocking complement receptor 3. I should say that desylation doesn't just do things to the microglia, it also does things to the neurons. So if you desylate the neurons, it makes them better targets for phagocytosis, as was first shown by Harold Neumann's lab, I think. And that's at least partly due to sialic acid acting as a don't eat me signal, activating cGLIT receptors on microglia, but also because opsonins such as C1q bind better to desylated neurons. So because microglial phagocytosis may contribute to neurodegeneration, we've tried blocking a variety of different phagocytic receptors to test whether this prevents neurodegeneration both in vivo and in vitro. And that can work well for acute pathologies, but the issue is whether blocking microglial phagocytosis is going to work for chronic conditions such as neurodegeneration, because most of those phagocytic receptors also contribute to removal of amyloid debris and pathogens, such as bacteria. So this drew us to another receptor, the P2Y6 receptor, which I've already mentioned. It's preferentially expressed on microglia within the brain, or almost exclusively expressed by microglia in the brain. And it's activated by UDP, the nucleotide extracellularly, which can be released from stressed neurons. And this was first shown by Kazumi and Inui in Kyoto in a Nature paper in 2007, where they showed that stressed neurons in vitro and in vivo can release UDP to activate the P2Y6 receptor on microglia, which can cause microglia to phagocytose neurons. And for example, they're showing here that if you add UDP to isolated microglia, it causes them to increase the phagocytosis of, in this case, fluorescent beads. But there's a 10-fold increase in this phagocytosis of beads, which is then inhibited by this inhibitor of P2Y6, MRS2578, or by knocking down P2Y6 prevents this phagocytosis. So we tested whether UDP could induce the phagocytosis of live neurons by just adding them to glial neuronal cultures. And in fact, yeah, adding UDP to glial neuronal cultures results in neuronal loss. That's what's being quantified here. In fact, that's relatively quickly. It can occur within a few hours and is accompanied by microglial phagocytosis of neurons. And that can be prevented, that neuronal loss can be prevented by blocking the P2Y6 receptor with MRS-2578 or just removing the microglia. So UDP induces neuronal loss via microglia and that requires the P2Y6 receptor. That was done in primary cultures from rats, and this is also the case in cultures from mice. So this is UDP-induced neuronal loss in mixed neuronal cultures from mice, but that doesn't occur in cultures from P2Y6 knockout mice. LPS, as I've said, causes a slow progressive loss of neurons, and this is the quantifying neuronal density after 3 days. So LPS causes neuronal loss, which again is prevented by this inhibitor of P2Y6, MRS-2578, and that can prevent neuronal loss induced by a variety of inflammatory agents. What about in vivo? So injected LPS into one side of the striatum of rats, and that induces an inflammatory activation of microglia. This is looking at the microglia with isolectin IB4, which preferentially binds to activated microglia. And this is striatum injected with vehicle. So you only see activation of microglia along the needle tract, but with LPS you get inflammatory activation of the microglia. And this is also staining the stained striatum for with an antibody against NUN, which is a neuronal nuclear specific antigen. And in this case, one can see neuronal nuclear material within the microglia. But what we're quantifying here is 1 week later, the neuronal loss induced by LPS on one side of the striatum relative to the other. And that's prevented if you co-inject MRS-2578 to block the P2Y6 receptor. More recently, we've used a chronic endotoxin model by injecting mice with endotoxin on 4 successive days into the peritoneum. So it's a peripheral injection. And that's been shown to cause relatively specific loss of dopaminergic neurons in the substantia nigra. And that's what we're measuring here 2 weeks after the injection of the last injection of endotoxin into the peritoneum. There's a loss of dopaminergic neurons in the substantia nigra, and we didn't find neuronal loss in other areas that we looked at, such as hippocampus or cortex. So there's a specific loss of dopaminergic neurons in the substantia nigra pars compacta. But that neuronal loss is prevented in P2Y6 knockout mice. So in the absence of this engulfment receptor required for microglial engulfment of neurons, you don't get this LPS-induced neuronal loss. So this endotoxin model is kind of a model of inflammatory neurodegeneration, but it's also a model of Parkinson's because it's been shown that Parkinson's patients actually have an elevation of endotoxin in their blood. And this may be due to the early gut dysfunction and gut leakiness that is seen in Parkinson's patients. So what about beta amyloid? So amyloid, if you add it at nanomolar levels to cultures, causes a slow progressive loss of synapses in neurons. And this is measuring the neuronal density after 3 days after adding 250 nanomolar amyloid beta. And again, this is prevented by adding MRS-2578 to block the P2Y6 receptor. It's also prevented by knockout of P2Y6 receptor. In vivo, we injected beta amyloid into the ventricles of mice, and 3 days later, we quantified the uptake of neuronal nuclear material into microglia. So this is a section of it through the hippocampus with the microglia stained green with IBA1, anti-IBA1 antibody coupled to a green fluorophore, and neurons stained with antibodies against NUN stained red. And occasionally you'll see microglia which have yellow within them, which indicates the uptake of NUN into the microglia. So that's the uptake of neuronal nuclear material into the microglia, again, here in this microglia and this microglia over here. So 2 weeks after injecting, this was 3 days after injecting a beta into the ventricles of mice, there was a large increase in the percentage of microglia that contained NUN internalized within them, both in the hippocampal CA3 region and CA1 and prefrontal cortex. And that's entirely prevented in the P2Y6 knockout mice. So in the absence of this engulfment receptor, there's no A beta induced phagocytosis of neurons. And 2 weeks later, we looked for neuronal loss in prefrontal cortex and in hippocampus, and there was a loss of neurons in prefrontal cortex and hippocampal CA3 and CA1, and that was prevented in the P2Y6 knockout mice. So in the absence of the engulfment receptor, there's no A beta induced neuronal loss. When we then went on to look at mouse brain function in terms of learning and memory, and we used novel object recognition to measure that. That's basically putting two objects into the mouse's cage, one of which is novel, and if they learn and remember that, then they spend more time with the novel object. And in this case, what we're seeing is about 75% of the time they spend with a novel object. But 2 weeks after injecting the A beta into the ventricles, there's a loss of learning and memory, so they're more or less down at chance whether they interact with a novel or non-novel object. So it means they have a deficit in learning and memory. Whereas the P2Y6, in the P2Y6 knockout mice, there's no A beta induced neuronal loss. There may be some decrease in the absence of A beta, but it's not significant. But in the P2Y6 knockout mice, there's no A beta induced memory loss. So the loss of the P2Y6 is sufficient to prevent A beta induced memory loss. What about tau? So Vilma Boratate's lab has shown that extracellular tau can cause slow progressive neuronal loss in these cultures due to phagocytosis. And Katerina Babushenko from Vilma's lab came over to Cambridge and replicated this in our cultures. So you get with extracellular tau, this does various things to activate the microglia, and you get a slow progressive loss of neurons, and that's prevented in P2Y6 knockout mice. Similarly, if you inhibit P2Y6 with MRS-2578, you can prevent the tau-induced neuronal loss. So we want to use a chronic model of neurodegeneration, and we used P301S tau mice. So P301S tau is a variant or mutation variant found in frontal temporal dementia and can cause frontal temporal dementia. But when genetically put into mice, it gives you a model of tauopathy, which develops at different rates with different genetic background. But in this genetic background, you get some neuronal loss at 7 months and some memory loss. So, but it is a fairly subtle neuronal loss, which we measured in perirhinal cortex and in motor cortex. So there is some loss of neurons in these two areas, and that's reduced if you cross these mice with P2Y6 knockout mice. So there's a reduction in the neuron loss, but there's full protection against the deficit in learning and memory induced by this model of tauopathy. So again, we're using novel object recognition, and the wild type mice with this background have spend 85% of their time looking at the novel object, but at 7 months, the P301S tau mice have a deficit in learning and memory, but crossing that to P2Y6 knockout prevents that deficit in learning and memory almost completely. I should say that in these mice, they have a fairly, at later time points, they have a fairly severe spinal pathology, which wasn't affected at all by crossing to P2Y6 knockout. We don't know the reason for that. It could be that the pathology is kind of stronger in the spinal cord, or that it involves a different mechanism of neuron loss. But it did prevent neuron loss in the brain and the memory loss. But because the neuron loss was pretty subtle in this model, we were interested in whether P2Y6 may also be affecting synaptic loss. So initially, we just looked at whether P2Y6 has any effect on the phagocytosis of isolated synapses, that is synaptosomes. And in pure microglial cultures from rats, we showed that inhibiting P2Y6 with MRS2578 reduced phagocytosis of synaptosomes, and in mice cultures, the knockout of P2Y6 reduced uptake of synaptosomes. To use a somewhat more intact model, we used the glial neuronal cultures, which developed synapses and synaptic puncta, which we measured, and used low-dose LPS. So at low doses of LPS, you can get a slow progressive loss of synapses with no significant loss of neurons. And this is what we're quantifying here with antibody against synaptophysin, which is a presynaptic marker. So we're quantifying these puncta, and the LPS induces a significant loss. But in cultures from P2Y6 knockout mice, there's no significant loss. Again, then in the absence of LPS, there may be reduced synapses, but there's no, although that's not significant, but there's no LPS-induced loss of synapses in the P2Y6 knockout. What about in vivo? So we used an aging model. It's not really a model, it's just natural aging of mice to 17 months of age. Results is known to cause synaptic loss, and there's a variety of evidence that involves microglial phagocytosis, and it also results in memory loss. So we looked at whether we could see changes in microglial phagocytosis of synapses by staining for synaptic puncta using an antibody against vGlut1, which is a presynaptic marker. And in red, you're looking at staining for vGlut1 in, I think this is a somatosensory cortex. And then we're staining for microglia with antibody against IBO1, and we're staining for lysosomes with an antibody against CD68. So what then we've got quantified is the uptake of, or the presence of synaptic puncta within the lysosomes of microglia. And this is a surface rendering of the microglia, with a surface rendering in blue of the lysosomes within the microglia, and a surface rendering of the synaptic puncta within the CD68 staining lysosomes. We quantified this in the young, which means 3-month-old mice, and the old, which means 17-month-old mice. And surprisingly, there was a large increase in the what we could measure as the presence of synaptic puncta within microglial lysosomes. But this was completely absent in the P2Y6 knockout mice, again indicating that P2Y6 may be contributing or regulating microglial phagocytosis of synapses. And we then quantified the density of synapses in cortex and hippocampus by looking at co-localization of a pre- and postsynaptic marker using vGlut1 as a presynaptic marker and HOMA1 as a postsynaptic marker, and showing that with age there's a loss of synapses, but that is reduced in the P2Y6 knockout mice. Again, in hippocampus, there's a loss of synapses, which is reduced in the P2Y6 knockout mice. And we then tested learning and memory in these mice by novel object recognition again. And again, the young mice spent 75% of their time with a novel object, but at 17 months of age, it's more or less down to chance whether they interact with a novel or non-novel object, indicating a deficit in learning and memory, which is prevented in the P2Y6 knockout mice. Again, the young mice may have reduced learning and memory, but it's not significant, but the aging itself causes no significant loss of memory. So knockout of P2Y6 is preventing aging-associated memory loss, potentially by blocking microglial phagocytosis of synapses. So in summary, I've told you that microglial phagocytosis may contribute to neurodegeneration, and blocking the P2Y6 receptor prevents microglial phagocytosis of neurons and neurodegeneration induced by inflammation, amyloid beta and tau, and also may prevent synaptic and memory loss with aging, and therefore may be a good target to prevent excessive neuronal loss in neurodegenerative diseases and potentially aging, although there may be other targets that are good targets to block microglial phagocytosis. So again, the main people involved in this work were Stefan Milde, Mar Puig-Delaval, Anna Villalta, David Allendorf, Claire Butler, Jake Dundee, and Alma Popescu. But there are a lot of other people that were involved over the years, and I probably not included all of them, but some of them are listed here. And this work was funded by either by the MRC, BBSRC, ARUK, European Union, or Wellcome Trust. So that's it. Thank you very much for listening.